You know, when you think about what you, a tree, and some tiny little bacterium all have in common, it's pretty wild. Turns out all life is built from this surprisingly small set of molecular blueprints. But here's the real secret. The magic isn't just in the ingredients. No, it's in the architecture, the exact three-dimensional shape that lets these molecules build and run the incredible machinery of life.
So how are we going to tackle this? First, we'll get to know the cell's main molecular players. Then, we're going to see why 3D shape isn't just some minor detail. It's literally everything. We'll find out how a tiny molecular flip is the secret to how you see. We'll explore the strange handedness of life. And we'll wrap up with the amazing story of the scientist who first figured all this out.
All right, let's dive right in. We're going to shrink ourselves down, go inside a cell, and just check out the basic parts list for making a living thing.
So take a look at this chart of a simple little E. coli cell. First thing you see, it's mostly water, right? But that other 30%, oh, that's where all the action is. It's almost entirely made of just a few types of huge molecules called macromolecules. These are the real engines and the actual building blocks of the cell.
And here they are, our four main players. You've got your proteins, they do most of the actual work. Polysaccharides, they're for energy storage. Then you have nucleic acids, like DNA, that's the blueprint. And lipids, which form all the protective walls and membranes. And what's really cool is that the first three are all what we call polymers, just these massive chains built from smaller, repeating molecular bricks. Okay, so we know the ingredients, but a list of atoms is kind of like a list of building materials from the hardware store. It doesn't tell you what the house actually looks like, right? To really get how these molecules work, we have to see them in 3D. And this whole science of 3D arrangement is called stereochemistry. And trust me, it's the key to everything. So I gotta ask, is a simple chemical formula really enough? Let's take a small amino acid. This one's called alanine. Yeah, its formula tells us what atoms are in it, but it gives us absolutely zero clues about how they're actually arranged in 3D space. And this slide right here shows you exactly why that 3D view is so critical. On the left, you've got the ball and stick model. Think of it like a skeleton, showing us the exact angles and connections between the atoms. But on the right, that's the space-filling model. That shows us the molecule's actual surface, the shape it really shows to the rest of the world. It's the same molecule, but these two pictures are telling us very different parts of its story. Now, this fixed 3D arrangement has a specific name. We call it a molecule's configuration, and you should think of this as being basically set in stone. You can't just twist it or bend it into a new shape. To change a molecule's configuration, you have to literally break chemical bonds, pull it apart, and rebuild it. You'd be creating a totally new thing. So let's look at a real-world example of this, one that involves a specific type of configuration caused by these really rigid double bonds. It's kind of like a tale of two molecular twins that look almost identical, but they have totally different personalities. Say hello to maleic acid and fumaric acid. They have the exact same formula, but they're what scientists call cis-trans isomers. See that double bond right there in the middle? It acts like a rigid bar. It locks the whole structure in place. So in the cis version, the bulky groups are stuck on the same side. But in the trans version, they're on opposite sides. That one tiny difference in shape gives them completely different chemical properties. And get this isn't just some drawing in a textbook. This exact thing is happening in your eyes millions of times a second. The very first physical thing that happens when you see something is a molecule called 11-cis-retinol getting zapped by a photon of light. In an instant, that energy makes it flip into its all-trans form. That single, tiny shapeshift is the trigger for the entire nerve impulse that your brain then calls sight. Amazing, right? Okay, now let's look at another type of molecular shape, one that's even a little bit weirder. It all starts with something called a chiral center, and it gives molecules a property that you use every single day without even thinking about it: handedness. A chiral center sounds complicated, but it's not. It's just a carbon atom that's attached to four totally different things. And because of this asymmetry, you can actually build the molecule in two different ways that end up being perfect mirror images of each other. And handedness is the perfect way to think about it. Seriously, look at your hands right now. They're mirror images of each other, right? They have all the same parts, a thumb, four fingers, but try to lay your left hand perfectly flat on top of your right hand. You can't. They just don't line up. They are fundamentally different in their 3D shape. Well, molecules with a chiral center are exactly like that.
Now, these special mirror image twins have a fancy scientific name. They're called enantiomers. Just think of them as one specific type of stereoisomer. Remember, stereoisomer is just the big umbrella term for any molecules that have the same formula but a different 3D shape. So this whole mind-bending idea of molecular handedness wasn't just figured out on a chalkboard. It was actually discovered in this brilliant and incredibly careful experiment by the one and only Louis Pasteur. So the story starts back in 1843. A young Louis Pasteur is studying something called tartaric acid. It's basically the crystal gunk that gets left over in wine casks. And as he's looking at these little crystals under a microscope, he notices something really tiny and strange that nobody else had ever paid attention to before. He realized there weren't one, but two types of crystals, and each one was a perfect mirror image of the other. So with what must have been just incredible patience, he sat down with a pair of tweezers and physically separated the left-handed crystals from the right-handed ones, one by one, into two piles. Then he dissolved each pile in water and shined a special kind of filtered light through each solution. And what he found was incredible. The solution with the lefty crystals bent the light to the left. The righty solution bent it to the right, by the exact same amount. He had physically proven the molecule's handedness. This was a monumental discovery. I mean, it was the first real proof that molecules have these specific 3D shapes, and that life itself is built on what we call stereospecificity. All those proteins and enzymes we talked about earlier, they are built to recognize only one-handed version of a molecule. A right-handed key won't work in a left-handed lock, and a left glove just will not fit on a right hand. Biology is that precise. And this brings us to this final, kind of mind-bending question. Life on Earth, for reasons we still don't totally get, decided to build all of its proteins from exclusively left-handed amino acids. So why that way and not the other? Could a mirror image version of life exist somewhere else out there in the universe? It's a pretty deep question that goes right to the heart of the secret architecture of life itself.